All right, hi everybody. I'm back for one more video. So what I'm going to talk about today is how to make a Punnett Square. This is going to go with part 2 of your lab right here, where we talk about doing Punnett Squares and being able to read them and make them and find out ratios of their offsprings of some parents. So how about we get started? So we're going to take this cross, and when we look at a cross like this, we're looking at the genotypic cross right here. So in this cross, we have a homozygous dominant genotype. So we know it's homozygous dominant because homo means the same and dominant is the uppercase letters. This is a homozygous recessive genotype. So it's homozygous because it's the same, and it's recessive because it's the lower letters. So if I were to ask you, what is the genotype and the phenotypes of the offspring of these parents? So one parent is big A, and the other parent is small a. What are the offspring of this cross, of these individuals? So in order to do that, you have to make a Punnett Square. And fun little Jeopardy fact, I don't think anybody cares about this, but I'll just tell you anyway. The Punnett Square is named after an English scientist called Reginald Punnett. So that's where that little history fact comes from. Anyway, so the way to make a Punnett Square is that on the sides of the Punnett Square, so up here and on this side, you put the gametes. And remember, if an individual is big A, that means they can make two gametes. They can either make big A or big A. And the homozygous recessive can either make small a or small a. So typically when we put a Punnett Square, the female gametes go up top and the male gametes go on the side. So I'm just going to put big A here, let's make it all nice looking, big A here, and over on this side is going to be small a, small a. And we might want to just make this a little more tidy. And bold everything. And of course, when you bold everything, all the formatting gets messed up. But that's how life goes in COVID. We have to be super patient. All right, we'll just get rid of that. So now that we have the maternal gametes up here and the paternal gametes down here, all you have to do is go into one of these squares and write what matches up. So see how in this square we have the big A and the small a, so that'd be big A, small a. In this square it would be big A, small a. In this square it would be big A, small a, and in this square it would be big A, small a. Each of these squares represents an offspring. So this is the genotype of a child. And in this case, the genotype, we would call that heterozygous. And that's really large, so we're going to change, oops, we are going to change the size to 12. Heterozygous. So when we talk about ratios, we could talk about how many of these offspring are big A. And so that equals 0. How many of them are big A, small a? And there's 4. And how many of them are small a? And that'd be 0. So 0, small a. So these are the ratios that you might see in your lab. So we're going to do another one. I'm going to clear the table. We're going to clear these. We're going to clear these. We're going to clear this. We're going to clear this. So what would happen, how would you do a Punnett square if the parents are both heterozygous? So we would put big A here. We would put small a, the recessive allele, here. We would put a big A here. And we will put a small a here. So what will go in this box? Well, this box corresponds to big A, big A. This box corresponds to big A, small a. This box corresponds to big A, small a. And this box corresponds to small a, small a. I think that was 20. So for this one, when we look at the phenotype, the genotypic ratios, we have 1 big A, 2 big A, small a, and 1 small a, small a. Okay, we did that Punnett square. And so from the Punnett squares, we can find out the ratios or the chance that the offspring are going to have certain genotypes and the phenotypes. So we'll do one more example, but this time we're going to bring phenotypes into it. And so I'm going to say, and we'll use the tallness, so we'll use t's. Big T, small t times small t, small t. So you might see something like this, and I'll say a heterozygous tall individual mates with a short individual. What percentage of offspring will be short? So for this, we have big T, small t. That big T, small t is the heterozygous tall. And this is the short individual. Why are they short? Because they don't have the dominant allele. So in our Punnett square, we're going to put big T, small t. Here we have big T, small t. So we do the, small t, small t. This small t comes from this individual. This big T, small t comes from this individual up here. So what is the genotype of this offspring? And it would be big T, small t. What is the genotype of this offspring? So you bring the small t down and you cross that with this small t, so that'd be small t, small t. What about this one? You'd have big T, small t, and this one is small t, small t. So is this individual tall or short? And the answer is that this individual is tall because it has the big T allele. Is this one short or tall? And this would be short. Why is it short? Because it doesn't have the big T. Is this one tall or short? Tall. And is this one tall or short? Short. So what is the percent of offspring that will be short? Well, 2 of them out of 4 will be short. And so if 2 out of 4 are going to be short, then that means that the answer should be 50%. So use this example, use this knowledge to answer all of the questions in part 2 of this lab. So all of these Punnett squares, all the problem solving, all the human traits, and then determination of sex in humans. All right, if you have any questions, ask your lab instructor. Have a good day.